Hello, dear students. Welcome to this new topic about genetic disorders. In this topic, you will be able to learn about the various human genetic disorders caused by alterations or aberrations in the structure and number of chromosomes. Generally, the number of somatic and gametic chromosomes of an organism ordinarily remain constant. This is a result of the extremely accurate mitotic and meiotic cell division. Definite types and numbers of genes are contained in each chromosome of a genome, and these are arranged in a definite sequence. But sometimes, due to a mutation or spontaneously without any known causal factors, variation in chromosomal number or structure may appear in nature. Chromosomal aberration may be classified into two broad classes, which are structural and numerical. Now let us look into the different ways by which chromosomes become altered and how these alterations cause some genetic disorders. The structure of the chromosome may be altered in two primary ways. First is by the change in the total amount of genetic material within a chromosome, and second by the rearrangement of genetic material within a chromosome. Variations in chromosome structure may result from chromosome breakage. Broken chromosomes tend to rejoin, and however, if there is more than one break, rejoining occurs at random and not necessarily with the correct ends. This results in structural changes in the chromosomes. Chromosome breakage may be a result of exposure to X rays, various chemicals, and can also occur spontaneously. There are four common types of structural aberrations or alterations. First is deletion or deficiency, and then duplication or repeat. Third is inversion, and lastly, translocation. The first structural aberration of the chromosome is deletion or deficiency. Deletion is defined as the loss of a chromosome segment. As a result, the affected chromosome is missing certain genes. However, if the centromere is deleted, the entire chromosome will be lost. There are two types of deletion. First is terminal deletion, and the second type is interstitial or intercalary deletion. Terminal deficiency or deletion results from a single break near the end of the chromosome. Now, if two breaks occur, a section may be deleted and an intercalary deficiency is created. In most cases, terminal deficiency appears less complicated, but majority of identified deficiencies are of the intercalary type of deletion. Remarkable genetic and physiological effects are generally produced in deletions. Most deletions are lethal when homozygous, since most genes are necessary for life, and homozygous deletions would result to zero copies of some genes. The next type of structural aberration of the chromosome is duplication. Duplication is defined as the occurrence of an additional chromosome segment as compared to what is normally found in the nucleus. The presence of a chromosome segment in a diploid organism in more than two copies per nucleus is called duplication. Consequences of gene duplications tend to be correlated with size. Small duplications are less likely to have damaging effects than comparable deletions. Also, more copies of a gene appear to be more harmful than just having a single copy. Four types of duplication are known: tandem duplication, reverse tandem duplication, displaced duplication, and translocation duplication. Tandem duplication occurs when the extra chromosome segment is somewhat located immediately after the normal segment in precisely the same orientation. When the gene sequence in the extra segment of the tandem is in the reverse position, it is known as reverse tandem duplication. Sometimes, the extra segment may be located in the same chromosome but away from the normal segment, and this is known as displaced duplication. Translocation duplication, on the other hand, occurs when the additional segment is located in a non-homologous chromosome. The next type of structural alteration is inversion. Inversion happens when a chromosomal fragment reattaches to the original chromosome, but in reverse orientation. Inversion being characterized by a rearrangement in the linear sequence of the chromosome usually results to the total amount of genetic material being unchanged. Generally, the genes within the inverted region are transcribed correctly, so most inversions do not have phenotypic consequences. The last structural alteration of the chromosome as a result of chromosome breakage is translocation. Translocation is the integration of a chromosome segment into a non-homologous chromosome. It can be classified as reciprocal and non-reciprocal. In a reciprocal translocation, which is the most common type, non-homologous chromosomes exchange fragments. Less often, a non-reciprocal translocation occurs. In this case, a chromosome transfers a fragment but receives none in return. In the previous slides, we talked about the different structural alterations of the chromosomes. Now let us move on to the second classification of chromosomal aberration, which is abnormal chromosome number. Ideally, the meiotic spindle distributes chromosomes to daughter cells without error. But there is an occasional mishap which is called non-disjunction, in which the members of a pair of homologous chromosomes do not move apart properly during meiosis I, or sister chromatids fail to separate during meiosis II. As a result, one gamete receives two of the same type of chromosome and another gamete receives no copy. This figure shows the event of non-disjunction in meiosis. Gametes with an abnormal chromosome number can arise by non-disjunction in either meiosis I or meiosis II. As mentioned previously, one gamete receives two of the same type of chromosome and another gamete receives no copy. The other chromosomes are usually distributed normally. If either of the aberrant gametes unites with the normal one at fertilization, the zygote will also have an abnormal number of particular chromosome, a condition known as aneuploidy. Aneuploidy may involve more than one chromosome. We most commonly see this resulting in a person with 45 or 47 chromosomes. Since there is a change in the number of chromosomes, then these individuals are no longer considered to be diploid or 2N. Aneuploidy being the loss or gain of one or more chromosomes can also be classified further as nullisomy, monosomy, trisomy, and tetrasomy. Nullisomy, or 2n-2, involves the loss of one homologous chromosome pair. Monosomy involves the loss of a single chromosome. When involving autosomes, monosomy may have severe phenotypic effects in animal species, but generally not in plants. Next is trisomy, or 2n plus 1, involves one extra chromosome addition. This is somewhat less severe than monosomy when involving autosomes. And lastly, tetrasomy, or 2n plus 2, involves one extra chromosome pair addition. Some organisms have more than two complete chromosome sets in all somatic cells. The general term for this chromosomal alteration is polyploidy. The specific terms triploidy or 3n and tetraploidy or 4n indicate three or four chromosomal sets respectively. One way a triploid cell may arise is by the fertilization of an abnormal diploid egg produced by non-disjunction of all its chromosomes. Tetraploidy could result from the failure of a 2n zygote to divide after replicating its chromosomes. Polyploidy is fairly common in the plant kingdom. In general, polyploids are more nearly normal in appearance than aneuploids. One extra or one missing chromosome apparently disrupts genetic balance more than does an entire extra set of chromosomes. For the next parts of my lecture, I will be talking about the specific human disorders. So at this point, let me talk about the human disorders caused by structurally altered chromosomes. Many deletions in human chromosomes, even in a heterozygous state, cause severe problems. One such syndrome, known as cri du chat or cry of the cat syndrome, results from a specific deletion in chromosome 5, thus otherwise called as 5p-. A child born with this deletion is severely intellectually disabled and has a small head with unusual facial features and has a high pitched cry that sounds similar to that of a distressed cat. The image on the right shows a patient with characteristic facial features of Cre du Chat, and the left image shows the deleted region on chromosome 5. Chromosomal translocations have been implicated in certain cancers, including chronic myelogenous leukemia or CML. This disorder occurs when a reciprocal translocation happens during mitosis of cells that will become white blood cells. In these cells, the exchange of a large portion of chromosome 22 with a small fragment from a tip of chromosome 9 produces a much shortened, easily recognized chromosome 22, which is called the Philadelphia chromosome. Prader-Willi syndrome or PWS is another genetic disorder caused by a structural alteration of the chromosome. PWS is a genetic disorder caused by a loss of function of specific genes on the long arm of chromosome 15. In newborns, symptoms include weak muscles, poor feeding, and slow development. Beginning in childhood, those affected become constantly hungry, which often leads to obesity and type 2 diabetes. So now let me discuss about the human disorders caused by a variation in the chromosome number. One aneuploid condition known as Down syndrome affects approximately 1 out of every 700 children born in the United States. Down syndrome is usually the result of an extra chromosome 21, so that each body cell has a total of 47 chromosomes. Because the cells are trisomic for chromosome 21, Down syndrome is often called trisomy 21. Down syndrome includes characteristic facial features, short stature, correctable heart defects, and developmental delays. The karyotype shows trisomy 21, which is the most common cause of Down syndrome. The child exhibits the facial features characteristic of this disorder. Although people with Down syndrome on average have a lifespan shorter than normal, most with proper medical treatment live to middle age and beyond. Many live independently or at home with their families, are employed, and are valuable contributors to their communities. Almost all males and about half of females with Down syndrome are sexually underdeveloped and sterile. The next genetic disorder is Patau syndrome, which results from trisomy 13. This syndrome may be characterized by mental retardation, growth failure, low set deformed ears, deafness, atrial and ventricular septal defect, microcephaly, cleft lip and palate, polydactyly, and etc. The image on the left of the slide shows some of the characteristic features of Patau syndrome. You can see significant microcephaly on the newborn with malformed ears and polydactyly or extra fingers. On the right of the slide is the karyotype of Patau syndrome. You can see the extra chromosome 13. There are some parental age correlation, but average age of parent is somewhat higher than for normal children. There are no evidence yet for maternal or paternal probabilities as source of the extra chromosome. The next genetic disorder is Edwards syndrome. Edwards syndrome is a product of trisomy 18. In Edwards syndrome, many problems are encountered and survival is less than 4 months. The occurrence of this genetic disorder is also associated with increased maternal age. This syndrome is characterized by growth failure, mental retardation, open skull sutures at birth, high arched eyebrows, low set deformed ears, and flexion deformities of the fingers. This image on the left shows a baby born with Edwards syndrome with deformed legs and flexion deformities both on the legs and fingers. The karyotype on the right shows an extra chromosome 18, which is the most significant feature of individuals with Edwards syndrome. Monosomy X, called Turner syndrome, occurs about once in about 2,500 female births and is the only known viable monosomy in humans. Turner syndrome is a product of non-disjunction during gametogenesis. Although these XO individuals are phenotypically female, they are sterile because their sex organs do not mature. When provided with estrogen replacement therapy, girls with Turner syndrome do develop secondary sex characteristics, and most of them have normal intelligence. The image of a girl on the left with Turner syndrome shows the distinct webbing of the skin at the neck region. Aside from this, girls with Turner syndrome also have cardiovascular abnormalities, hearing impairment, and rudimentary ovaries. An extra X chromosome in a male producing XXY occurs approximately once in every 500 to 1,000 live male births. People with this disorder, called Klinefelter syndrome, have male sex organs, but the testes are abnormally small and the man is sterile. Even though the extra X is inactivated, some breast enlargement and other female body characteristics are common. Affected individuals may have subnormal intelligence. Males with the syndrome have characteristic thin, long arms and legs, large hands and feet, underdeveloped testes, developed breasts, no facial hair, slow learner but not mentally retarded, sterile but not impotent. They can also live a fairly normal life with hormone supplements. So, this is the end of my discussion about genetic disorders. I hope you guys have learned something. Thank you for watching this video.